Hey there, micro friends. Welcome to another episode of Let's Talk Micro. As always, I'm so happy that you're here with me. And if this is the first time you're listening to Let's Talk Micro, well, thank you for choosing it as one of your podcasts. And hey, if you're listening to this, this week as this episode is being released, ASM Microbe is happening at Washington, D.C. So if you're there, if you're attending from June 4th through the 7th, and you see me, hey, say hi. And if you would like a Let's Talk Micro sticker, let me know because I'll definitely give you one. So stay tuned till the end so I can once again give the announcement about my live episode at ASM on June 6th. So let's go ahead and talk a little bit about the episode. Well, this is the second part of the hantavirus episode with Ryan Relich and Stephanie Seifert. And while on the first episode we started giving like a general overview of hantaviruses, on this one we talk about the Andes virus and the whole cruise ship situation that I'm sure you have seen on the news. And we talk about testing. We talk about the whole cruise ship situation. We talk about the concerns of the pandemic like thoughts and things like that. So it is a great episode. So let's go ahead and listen to it. Okay, well, thank you for that. And let's talk a little bit about the Andes virus and we've been hearing, as I said before, we've been hearing a lot of information through like news, social media. So just to kind of bring it all to one place, maybe you can start touching real quick on what happened here with this cruise ship, and then tell us more about this particular virus.
And so I think this is kind of a worst case scenario for this particular virus. So there's certainly been other events where this virus has had outbreaks, including a wedding where there's human transmission, and that's really the key feature of Andes virus among the hantaviruses. It's the only one where we have documented human to human transmission. And so that's why it's a big concern when you have an index case that's emerging on a cruise ship, on a cruise ship full of passengers from all over the world. And so there's a few challenges with this situation. One is that cruise ships are environments where pathogens tend to transmit very readily. Another such environment are hospital settings. So we say nosocomial transmission is something that we're generally concerned about, especially with respiratory viruses, but also with viruses like Ebola viruses, where you have a high incidence among hospital staff. I'd like to think that our experience with COVID-19, with SARS-CoV-2, has made us better prepared for dealing with acute respiratory distress and a respiratory virus in terms of people who are trained and capable to deal with a respiratory virus. It requires special handling and PPE. On a cruise ship, that's not likely to be the case. Cruise ships are typically not furnished with a full, hospital setting. And so to have it occur in the middle of the ocean, I think that this ship left Argentina on April 1st, if I'm remembering correctly. And then the index case passed away on April 11th. Somewhere, I guess, between it was Argentina to the Arctic and then to South Africa. So somewhere along that route, which is also very isolated on a cruise ship with, medical settings that just really aren't prepared for something this severe. It's in that way kind of a worst case scenario. So the concern was that we have the ship full of passengers who may or may not have been exposed, and then they want to go home, and so then we're sending them all around the world, and then it's the question of like, where is this virus going to get to, and how prepared are the hospitals and the clinics that are going to be dealing with um, these folks who may have been exposed, until we've cleared that window of time where we don't think that the virus is going to emerge in any of the people who might have been exposed. So the quarantine, I think, right now is really important. From there, it's we've had quite a few deaths that were associated early on with like this cruise ship before people were being evacuated to hospitals that could actually deal readily with these cases. I think there was a doctor even on the cruise ship who stepped in and tried to assist as best he could, and I think that doctor also contracted the virus. And this is something that we also see with Ebola viruses frequently, is that it's just very challenging when you're in that close of contact with somebody who is experiencing severe disease, to avoid getting infected yourself. So from that perspective, I'd say our critical thing right now is to continue monitoring the passengers, which have now been that left the cruise ship and are now at their respective home countries, kind of waiting out the period to make sure that nobody else gets sick. I think it's very unlikely that we're going to see more deaths as a result of this outbreak at this point. I think if anybody does start to show symptoms, that they're being monitored very closely, and that they're going to get the access to medical care that is needed to get them through. As Dr. Relich was saying earlier, that this is reversible, but there is a critical period of time where, especially if someone progresses to severe disease, because we don't have any specific therapeutics or treatments, it might just be that we need to get them hooked up to an ECMO to replace their heart and lung function until they have a chance to recover. And so I think that the outlook for the people who might have been exposed on this cruise ship at this point is very good. I think that the probability that we're going to have onward transmission is very low at this point, because the public health response has been very swift now, and I think the public in general are willing to tolerate things like, we need to just control the folks from the progression of this outbreak from this cruise ship is a very different thing from, we need to do countrywide lockdowns for COVID. Um, and so it's partly just how much of a public health response the public are willing to accept. And with a virus like Andes, it seems like folks are willing to say that we should have quarantine for the people who are coming off the cruise ship for some period of time, and that's an acceptable level of public health intervention and response. And I do think that's going to be enough to control the onward infection. Um, Again, with Synovial virus, we've never had any evidence of human-to-human transmission. With Andes virus, we have. And so that's the one that we really watch out for, in terms of, can this continue into a more prolonged outbreak. I've been asked a few times if this virus can have what we call spill back, if there's any risk of spill back in these other countries where now we have, patients actually being cared for, but also people who may have been exposed. And I think that the answer is, like, it's always a possibility, but I think in this case, it's a very low risk. Typically, when we aren't very efficient at transmission ourselves, the risk to animals around us is generally lower than the risk to other people, especially people who are interacting very closely with us. So things like MERS coronavirus, where you have a lower respiratory infection, and we have a low human-to-human transmissibility with that virus. I think it's unlikely that we're then going to be able to infect, an animal that may or may not be susceptible, like a dog or a cat, from a virus that we're barely shedding ourselves. So I think it's unlikely at this point. Obviously, with SARS-CoV-2, it's highly transmissible in humans. Clearly, we're shedding virus in a way that facilitates transmission. And we have had many cases documented of spillback for SARS-CoV-2. Right now, I think the risk of that happening with the Andes virus is very low.
Okay. And
Ditto.
Yeah, sorry, go ahead.
I said ditto.
Okay. And you answered one of my questions when it comes to with treatment, but from the lab side, right? So we have to talk about testing. So where are we with testing for this Andes virus?
So, Andes virus specific diagnostic testing cannot be done by commercial or hospital based clinical laboratories. That type of specific testing is really relegated to either research laboratories or public health labs like the Centers for Disease Control, or whatever the cognate of those facilities are in other countries. So testing options available for hantaviruses, for example, in the United States, and globally, there's generally serological test methods, nucleic acid amplification, and antigen detection via immunohistochemistry or immunocytochemistry. And serology is really the gold standard. Here in the United States, serologic tests, including IgM capture ELISAs and IgG ELISAs, are available at a number of commercial reference laboratories and also in areas where you see a high incidence and prevalence of hantavirus infections, like, for example, in California and New Mexico. Many clinical laboratories will also offer IgM or IgG testing because they are the states where you see a higher proportion of hantavirus pulmonary syndrome cases. But here in Indiana, for example, we would have to submit a specimen to a commercial reference lab, either Mayo Clinic Laboratories, ARUP, Quest Diagnostics, LabCorp, et cetera, et cetera. And so that testing for antibody-based detection of hantavirus infection, it is, again, the gold standard. However, it lacks sort of analytical specificity among the orthohantaviruses. And so in order to find out whether it's specifically Andes virus or Pumala virus or Sinombre virus or whatever your favorite orthohantavirus is, you would have to do something more specific as a follow-up. Generally, with viral serological tests, if you see a fourfold increase in IgG, you may just say, okay, it's whatever this virus is. But if you do want to know specifically what those antibodies have, what antigens they've been raised against, you would do something like focus reduction neutralization testing or plaque reduction neutralization testing, depending upon the type of virus that you're working with. And so hantaviruses, they're not known in the viral culture world for being excellent producers of cytopathic effect, like many other viruses, including SARS-CoV-2 are. So you do something called a focus reduction neutralization test, and you look for a fourfold difference in neutralization for the different viruses. And so basically what you do is you grow cell cultures and monolayers within reaction vessels of some sort of substrate, like a 24-well plate or a 96-well plate or a 6-well plate or something. And you mix a heat-inactivated volume of the patient's serum with known quantities of the different viruses in separate tubes. And if those antibodies that are present in the patient's serum identify those viruses, they will bind to them and neutralize the infectivity of those viruses. And so after you incubate the serum with the virus for a specific period of time, you'll add that to the monolayers of the cells. And then after a period of incubation, you'll add antibodies, or you can use fluorescence as a readout, whatever. And you'll look for a reduction in the focus of infection. So you can use, some sort of an immunocytochemical reagent to look for a color change. And those little color changes, those little spots, are your foci of infection. And so you have controls that you incubate along with it, where there's no antibody, and also specific antibodies. And so if you have an antibody that recognizes Andes virus, you'll see a reduction in the number of foci that are produced, because those foci going away means, or not appearing in the first place, means that the antibody neutralized the Andes virus. And so that's serology in a nutshell. And nucleic acid amplification testing is also useful. Again, it's not common, though. Generally, it's only public health laboratories that offer that to the community at large. Most clinical laboratories, even specialized reference laboratories, do not offer nucleic acid amplification testing. Most NATs that are performed for hantaviruses are in the form of real-time PCR, real-time RT-PCR, reverse transcription PCR. And generally, RNA can be detected from plasma or serum and a number of other body fluids, generally within 3 to 10 days after symptom onset. And so blood is collected, and possibly other fluids may be collected and can be then submitted for nucleic acid detection in a public health lab. However, that's not sort of the end-all be-all. So you can actually get clues as to whether or not a patient may have hantavirus pulmonary syndrome in any clinical laboratory that performs CBCs with differentials. There are hematologic manifestations that are characteristic of HPS. And in general, a patient's blood picture must satisfy four of the five following criteria. Number one is hemoconcentration. And hemoconcentration is indicated by an increased hematocrit. In addition to that, thrombocytopenia, neutrophilia, generally without toxic granulation of the neutrophils, so a neutrophilic leukocytosis, also a general myelocytosis, and circulating immunoblasts. You don't always see cases with circulating immunoblasts, but generally in cases where you do see the circulating immunoblasts, more than 10% of the lymphocytes may be blasts. In some case reports, upwards of 40 to 50% of the circulating lymphocyte-like cells are lymphoblasts or immunoblasts. And so, in general, laboratory criteria for diagnosis of hantavirus-specific disease is detection of hanta-specific IgM or a fourfold or greater rise in hanta-specific IgG, as well as a positive RT-PCR result, whether that's a real-time test or an endpoint PCR. And in cases of fatal disease, actually detection of hantaviral antigens within tissue that are collected postmortem. Those include lung, spleen, kidney, and other solid tissues. Again, there's not specific histopathologic lesions that you'll see, like cytopathic effects that are diagnostic of hanta pulmonary syndrome, but if you see lung tissue where you see this preponderance of intraalveolar edema, as well as some other sort of generalized pathologic features. And you also can do hantavirus immunohistochemistry and localize the viral antigens. Those are all very good clues that the diagnosis is going to be HPS.
I would like to see more PCR.
More PCR would be great. If more labs could develop that testing and implement it, that would be fantastic.
Yeah. I'll say also it's a reportable disease in the United States. So if you have or suspect hantavirus, then the CDC can get involved and confirm findings as well. So there is also a central repository for positive testing data with the CDC, which is, I think, from an epi standpoint, it's quite nice to have that resource because typically if you have an increase in cases in California, then you might also want to be looking in Washington State or New Mexico, et cetera. And luckily, at least with the hantaviruses, I do happen to know that those departments of health are very communicative and they are collaborative and they reach across those state borders. But in general, for infectious diseases across the United States, CDC has a role that we desperately need. So really need CDC to continue being a hub for centralized information for diseases like this.
There really is a good faith effort too among clinical laboratories to steward that information to the appropriate um, public health authorities and other things. And there are actually some clinical laboratories that are now starting to publish, for example, commercial reference laboratories that are starting to publish on their websites information about sort of the incidence and prevalence of various infectious diseases like pertussis and respiratory diseases, GI diseases, so on and so forth. And it's those laboratories that get specimens from all over the country or even all over the world that really hold in their hands the data that is very useful for informing public health decisions, infection control practices, ring vaccination events if they need to be held, so on and so forth. So I think too, the continued collaboration of hospital and clinical laboratories with public health labs is absolutely essential.
So thank you both for your time. And as we start wrapping up the episode, and, we have shared a lot of information. But before I ask the last question, which is always meant to end things to a lighter note, we have shared a lot of good science, so it's always nice to end on something light. But before I do that, um, do you have any final thoughts about this? Anything that you want to share or thinking about for the listeners?
Yes, I do. My final thought is um, and I'm not up on a pedestal when I'm saying this, but we need to continue supporting and financing research into these types of things. The type of work that Dr. Seifert does is absolutely critical to getting a handle on these things. I mean, as a clinical laboratorian, I kind of see, have a front row seat to the early detection of these things whenever they spill over into the human population. But understanding the transmission dynamics from where these agents come from in nature is absolutely critical. And so I'm always a big proponent of funding science. And so Dr. Seifert's laboratory does exceptional science. And so we need more of that.
I want to kind of piggyback on that with the diagnostic need. Having infectious disease doctors to be able to keep track of what viruses are causing these infections is really critical. And I think oftentimes there's such a focus on patient care, which is absolutely necessary. We need people to be able to focus on patient care. But also identifying trends requires that we get an accurate diagnosis. So it's not just about saving that patient, but it's also about getting us the clear information on which pathogens we need to worry about so that we can also prevent disease in the future and save more lives beyond just that initial patient. And so that's really critical too. And in Argentina, I think we could have kind of foreseen that something like this could have happened because this season has been really a bad season for hantavirus pulmonary syndrome in Argentina. So preceding the cruise ship outbreak, there had been over 100 cases this year. And so we knew that this was a problem in Argentina this year. This is more than double the cases that they kind of anticipate happening on an annual basis. And then it's only when it spills on to this cruise ship that we start to get the global kind of involvement and global view that this outbreak is happening. And it's those 11 cases have had more press, I think, than Andes virus ever has. And that's really something we can work on, I think, as a global community that, disease is happening on a local level or something that we should keep an eye on before they become a global outbreak. And that's true for Mpox, right? It had been showing signs of increased human-to-human circulation for 5 years prior to the 2022 pandemic. So I think this is a pattern that we should kind of recognize now and then try to intercept earlier.
Well, thank you for that. And so let's go ahead and go to the last question that I have. As I said, to keep things in a lighter note. So we all have that favorite or least favorite bug. It can be any number of reasons. People either like it. The answer that I tend to get the most is the person worked with it in school. So it has that significance, about it. But it could be anything visual, anything like a mechanism, even a smell. Bottom line, we all have that favorite bug. So I'll start with you, Dr. Seifert. What is your favorite bug?
Um, so when you say bug, because I am also a taxonomy nerd like Dr. Relich, I'm going to assume that you're talking about hemipterans, and I'm going to say a tree hopper. I'm a big fan of tree hoppers. I actually, in general, really love insects a lot. So I'm a big fan of katydids too. I can't get enough katydids when I'm doing field work, and that's one of the more delightful parts I think of my job is when I get to travel to these remote places and we're setting up sampling at night, either for bats or rodents, and then because we're the only light source around, we get the most amazing diverse insects. So I like true bugs a lot.
What about favorite virus? Do you have a favorite virus, Steph?
I'd say a favorite virus. I have a lot of viruses that I'm really curious about. So a Sonoma virus has been a focus of my attention for many years now, and I'm also just so interested in the evolutionary history of what we call OC43, which is a human seasonal coronavirus, but we also call that bovine coronavirus and canine respiratory coronavirus and giraffe coronavirus. So basically any animal that this virus has been found in, we call it, that animal coronavirus. And so I'm really interested in the evolutionary history of beta coronavirus. Gravidinus is the proper species name for that virus, but we think it could have been the cause of what's now called the Russian flu. Might not have been the flu at all, but instead the introduction of beta coronavirus gravidinus into humans, either directly from a rodent reservoir or indirectly via cattle. And that virus has since jumped into equines, and then it adapts. It jumps into swine, and then it adapts. And I just think that virus is so interesting. And possibly, if we can understand how that virus has emerged and then evolved and kind of persisted in different animal populations, that'll tell us something about what we can anticipate from SARS-CoV-2. So that's my current fascination is beta coronavirus gravidinus. How about you?
I have several. It depends upon DNA virus versus RNA virus, and I also have favorite bacteria and fungi and parasitic protozoa. But so as not to keep you all on the line for the next 45 minutes, my favorite RNA viruses as of right now are bourbon virus and heartland virus. So yeah, I like them quite a bit. I like tick-borne viruses, and I've got my bat here, and other bats around the room. I like a lot of bat reservoir viruses, henipaviruses, parahenipaviruses, lysoviruses. So yeah, I've got lots.
Perfect. Did you say bourbon?
Bourbon virus, yeah, it's named after Bourbon County, Kansas, where it was originally discovered.
Hmm.
I have my little red river hogs here. I don't know if you can see them. But when we were in Bikoro in Congo during an Ebola outbreak, we were trying to figure out which animal that virus emerged from. And we actually had a red river hog skull. And we tried to isolate or see if we could amplify nucleic acids from Ebola virus out of a tooth that we extracted from a skull that was from a carcass that the index case had consumed. So it's always interesting and fun field stories.
Perfect. Okay, well, this has been great, and and I hope the listeners enjoyed. I definitely did, and I said this to Dr. Relich before when I had him on, and now with you, Dr. Seifert too. It felt like a nice lecture where I was just listening here and have to keep in mind, hey, I'm doing a podcast and I have to keep track of the questions and the time. So thank you so much for both of you for taking the time to be in Let's Talk Micro.
Absolutely, thanks for having us.
Yeah, thanks so much for having us.
Well, there you have it. I hope you enjoyed the episode. As always, I enjoy sharing this information with you. And there's so much information out there, so make sure you get it from legitimate sources. And in this community, there's a lot of scientists putting information out there, good information, accurate information. I hope this episode helps as well. So be responsible with the use of information. Hey, and once again, if you're listening to this and you're attending ASM Microbe in Washington, D.C. from June 4th through June 7th, please check out my live episode. It's going to be at the ASM studio on Saturday, June 6th from 3 p.m. to 3:30. And it is titled, What Can Whole Genome Sequencing Realistically Tell Us About Resistance? And for this episode, I will be joined by Dr. Nathan Littebor, and Andrea Princey is going to be co-hosting with me. So if you're there, please check out the episode. I would love to see you there, showing your support. So June 6th from 3 to 3:30 p.m. at the ASM studio. So when you go to the exhibitors area, check out the ASM area and we will be there. So I hope to see you there. Okay, so as always, we are on every podcast platform. So if you go to Apple, Spotify, Amazon Music, GoodPods, you can find us right there. So please subscribe to the podcast, download episodes, and if the app allows you to do so, please leave a review. Thank you so much for the support. And you can find us on socials on Instagram, TikTok, YouTube, Blue Sky Social as Let's Talk Micro. We're on X as Let's Talk Micro 1, LinkedIn as Luis Plaza. And of course, there's the website, letstalkmicro.com, where you can find episode updates, you can find licensing information, educational resources, and more. And there's an email address, which is letstalkmicro@outlook.com. So either via social media, via email, you can reach out with any suggestions, any feedback. They are always welcome and appreciated. And if you want to support us in any way, you can do so via Venmo or via Buy Me a Coffee, KOFI, both links available on the show notes. And friends, You know that in microbiology and infectious diseases, something that is very difficult is learning about antibiotics. So, if you would like to learn more about antibiotics, please check out www.learnantibiotics.com and the Learn Antibiotics book available on Amazon. These resources include cheat sheets, practice tests, games, and more, and they are being used by thousands of people worldwide and may be helpful for you or your colleagues. And as always, these are some great resources from Dr. Timothy Gauthier, a very passionate pharmacist. And as you know, the Let's Talk Micro podcast is supported by the American Society for Microbiology. To learn more about microbes, visit asm.org. And that, micro friends, is the end of this episode. I hope you enjoy learning about the Andes virus. As always, I enjoy sharing this information with you. If you're an ASM microbe, hey, if you see me, say hello. I'm looking forward to meeting you and connecting with you. So say hi, and hey, if you have time, support the episode. I would love to see you there. Please continue bringing that passion to what you do. It's so important. You do such great work. And as always, thank you for the support. As always, stay motivated, stay safe, and of course, continue talking micro. Until the next time. Bye.